Hi, I'm Dr. Jonathan Whitlock. I'm a group leader at the Cobley Institute for Systems Neuroscience in Trondheim, Norway. I have a background in studying spatial navigation and spatial computations. My group has recently shifted its focus now to focus more on action perception, action understanding, and action planning. My research group began about 3 years ago, and I knew that one of the changes I wanted to make in my line of research was to use calcium imaging in freely moving animals simply because of the very large number of cells that you can get at a given point in time. In one of the lines of research going on in the lab now, we've found that there are in fact subsets of cells in the rodent parietal cortex that are involved in executing certain actions, but what's quite new is we found that there are subsets of cells that are involved in representing another animal's actions. We want to do more population level recording while the animals are switching between performing and observing various motor behaviors. One of the outstanding questions is, if you have a group of cells that's responsive to ongoing behavior, are those cells wired differently than cells which respond to observed behavior? So are the cells which are responsive to performance and observation connected with other areas in the same way? One of the strategies that we're going to use to hopefully functionally dissect the action planning and action perception networks is to image cells on the basis of the areas that they project to. You can do this using viral targeting strategies. This is an advantage that you have when you're doing calcium imaging that you just really can't implement very effectively when you're doing, for example, tetrad recordings. So this is an approach that you can pull off very nicely using the Invista system and in vivo calcium imaging. That's very hard to do electrophysiologically. To learn more about my line of research involving action planning and action observation in the cortex, you can visit my lab's website.